morning, everyone. My name is Pearl Alfami D. Maturan, and I'm the first one who will going to report. So, genes and human genomes. The human genome is a complete set of nucleic acid sequences for humans, encoded as DNA within 23 chromosome pairs in cell nuclei, and in a small DNA molecule found within individual mitochondria. Chromosomes. Each chromosome is made of protein and in a single molecule of deoxyribonucleic acid or DNA that pass from parents to offspring. DNA contains the specific instruction that can make each type of living creature unique. Gene. A gene is a basic physical and functional unit of heredity that made up of DNA. Some genes act as instruction to make molecules called protein. DNA is the chemical name for the molecule that carries genetic instruction in all living things. The DNA molecule consists of two strands that wind around one another to form shape known as double helix. Each strand has a backbone made of alternating sugar or deoxyribose and phosphate group. Genetic disorder. Genetic disorder occurs when a mutation or a harmful change to a gene, also known as pathogenic variant, affects a person's genes or when a person have the wrong amount of genetic material. Some genetic disorders are serious. The children who have them are extremely sick and cannot survive after birth. Single gene disorder. Even though these diseases are primarily caused by a single gene, several different mutations can result in the same disease but with varying degrees of severity and phenotype. The DNA changes affect the product that the gene code, usually a protein, that causing it to be altered or missing. For many single gene disorder, the genetic basis is well understood, and the disease-causing gene variants can be identified by genetic testing. People are tested for two main reasons: to find out if they have a particular genetic disorder or to find out if they are a carrier. In first, making a diagnosis testing a person's disorder based on their symptoms. Second, identify carrier status. Autosomal gene disorder. In an autosomal recessive disorder, one mutated gene is inherited from each parent. Each parent carries one mutated gene and one normal gene for the disease, although the parents do not have the disease. And an autosomal recessive disorder means that the gene in question is located of one of the autosomes. Two carriers have a 25% of chance of having an unaffected child with two normal genes, a 50% chance of having an unaffected child who is also a carrier, and a 25% chance of having an affected child with two recessive genes.
Hi everyone, once again good morning. I am Joanne Ambatak and I'm one of the presenter in Group 7. So now let's tackle about the X-linked disorder. X-linked disorder is originated from the genes of sex chromosomes, in which it is caused by a variance in genes on the X chromosomes. So here in the picture is the X-linked recessive. In the first side of the picture is an affected father and carrier mother. They have 4 children, which is 2 sons and 2 daughters. If the mother is a carrier, there are 25% chances kamao ni ang result sa iyang pregnancy, which is an affected son, carrier daughter, affected son, and unaffected daughter. So in the other side of the picture is affected ang father, ang mother ang unaffected. They also have 4 children, which is 2 sons and 2 daughters. Their 2 sons is unaffected, and only the 2 daughters is carrier. The next slide is example of X-linked disorder. So first is the hemophilia. Hemophilia is a blood clotting disorder that caused by a mutation. The next example is color blindness. So color blindness is you see colors differently than most people, and most of the time Color blindness makes it hard to tell the difference between certain colors. Like for example is the red-green confusion. This type of color blindness is having difficulty seeing shades of red, green, and yellow. Chromosome disorder. Chromosome disorder might be an excess or deficiency of the whole chromosome, chromosome rearrangement, and contiguous gene syndrome. It is a set of chromosomes as seen under microscope is known as karyotype. Any deviation from the normal karyotype is known as chromosomes abnormality. A chromosomal disorder is a syndrome that characterized by malformations or malfunctions in any of the body systems, and by that it can cause abnormal chromosomes number or constitution. If one of our chromosomes will malfunction or malformation, and if there are changes in number of chromosomes, it will result as a chromosomal disorder. We have three examples of chromosomal disorder. One example of it is Klinefelter syndrome. Klinefelter syndrome is born with an extra X chromosome. Instead of the typical XY chromosome in men, they have XXY. So their condition is sometimes called XXY syndrome. There is no cure, but it can be treated. So Klinefelter syndrome is a genetic condition that results when a boy is born with an extra copy of X chromosomes. This syndrome is affecting males and it isn't diagnosed until adulthood. So, as we all know nga, pag male, ang chromosomes niya is XY. And if female, XX ang chromosomes. But kani siyang Klinefelter syndrome is naduha iyang X, so which is nagay changes, nagay different sa iyang pag-grow sa iyang body. Dili siya same sa uban laki nga nya na ang pag-grow sa kuan sa ilahang body. Kani siya naa siya mga changes or mga signs. Like for example, yung mga signs is a taller, less muscular body than males their age, broader hip and longer legs, larger breasts, weaker bones, a lower energy level, smaller penis and testicles, delay in puberty or go a partial amount, less facial and body hair following puberty. So, buo na siya iyang mga signs nga nakalahi gyud siya sa uban lalaki. Second syndrome is the Turner syndrome. 1/5,000 females, gonadal dysgenesis, disappearance of eggs, sexual immaturity, amenorrhea, infertility, short stature, somatic abnormalities, something wrong with the second X in some cells, missing an X chromosome on 23rd pair. So, in Turner syndrome, it affects only in females. It results when one of the X chromosomes is missing or partially missing. Turner syndrome can cause a variety of medical and developmental problems, including short height, failure of the ovaries to develop, and heart defects. Females with Turner syndrome grow more slowly than other children. The next slide is the sample picture of the Turner syndrome.
Down syndrome or trisomy 21 occurs because of the extra copy of chromosomes 21 which can cause the body and brain develop differently than a child without the syndrome. Down syndrome is caused by the trisomy 21. The person has 3 copies of chromosome 21 instead of the usual 2 copies in all cell. This is caused by abnormal cell division during the development of the sperm cell or the egg cell. It is also one of the common genetic birth defects. Some physical features Down syndrome include a flattened face, especially the bridge of the nose, almond-shaped eyes that slant up, a short neck, small ears, a tongue that tends to stick out of the mouth, tiny white spots on the iris of the eye, small hands and feet. Pato syndrome or trisomy 21 caused by having an additional copy of chromosomes 13 in some or all of the body of cell. Pato syndrome is a serious rare genetic disorder caused by having an additional copy of chromosome 13 in some or all of the body of cell. It is also called trisomy 13. Each cell normally contains 23 pair of chromosomes which carry the genes you inherit from your parents. Pato appears to affect more females than males, most likely because male fetuses do not survive until birth. Individuals with trisomy 13 often have heart defects, brain or spinal cord abnormalities, very small or poorly developed eyes, extra finger or toes, an opening in the lip or without an opening the roof of the mouth, and weak muscle tone. Edwards syndrome, caused by the presence of all part of extra 18 chromosome. The majority of people with this syndrome die during the fetal stage. Infants who survive experience serious defect and commonly live for short life. So, trisomy 13, also known as Edwards syndrome, is a genetic disorder that affects baby and can often be diagnosed before birth. A fetal ultrasound during pregnancy can show features that are suggestive of trisomy 18. Most types of Edwards syndrome cases are not hereditary, so they cannot be prevented. To date, there is no scientific evidence as to what a parent could done to cause or prevent trisomy 18 in their baby. So this is a chromosomal error. This is considered to be random. Affected individuals have heart defects and abnormalities of other organs that develop before birth. Other features of trisomy 18 include a small abnormality shaped head, small jaw and mouth, a
Hello
clenched
everyone, I
fist
am
with
Angel
overlapping fingers.
Madronero and I am going to discuss about the topic multifactorial inheritance and gene therapy, including its benefits and disadvantages. So let's first tackle about the multifactorial inheritance. So what is multifactorial inheritance? It is called as complex or polygenic inheritance caused by the combination of environmental factors and mutation in multiple genes. When we say polygenic or complex inheritance, it is consists of many different connected parts that contributed to multiple genes. And the main factor of multifactorial inheritance is the genes. But there are some other factors that include such as lifestyle and nutrition, birth defect, or chronic illness. And when we say chronic illness, it is the human health disease that is long-lasting effects, 1 year or more, in which required ongoing medical attentions, just like high blood pressure, arthritis, diabetes, cancer, obesity, Alzheimer's disease, and heart disease. They are the diseases that need or required ongoing medical attentions. So the next one will be the gene therapy. So the gene therapy is a method of treating or preventing diseases or replacing or repairing or returning on or off genes of a patient's cells. So the gene therapy is the process or way of repairing disorders of genes caused by mutations through medical approach to prevent disease by correcting the underlying genetic problem. It allows the doctors to treat disorder by altering a person's genetic makeup instead of using drugs and surgery to produce the therapeutic effect or treatment of diseases. And there are two types of gene therapy: the somatic gene therapy and the germline or reproductive gene therapy. Somatic gene therapy doesn't alter the genetic makeup of an organism. It involves obtaining blood cells from a person with a genetic disease and introduce the normal gene into the defective cell. It also has the ability to introduce genetic material into the appropriate cell type or tissue to produce a therapeutic effect. And also, it involves placement of a human gene into a living person's somatic cells. It aims to cure a disease only in the patients but not in the patient's descendants. There are three types of somatic gene therapy. The first one will be in vivo. It is a genetic material, usually in the form of DNA. The second one will be the in situ. It consists of administration of the gene product to a specific site. The third one will be the ex vivo. It involves the genetic modification of cells outside of the body to produce the therapeutic factors and their subsequent transplantation back into the patients. And the next one will be the reproductive gene therapy. the alteration of genetic makeup of the offspring by targeting the abnormal genes in the parent's sex cells. So the reproductive gene therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. It allows insertion of gene into the reproductive cells or into the genome. The next one will be the benefits of gene therapy. The benefits of the gene therapy is to offer cure to several diseases like cancer, asthma, diabetes, heart diseases, as well as the hereditary diseases. And when we say hereditary diseases, it is a genetic disorders occur when one or more genes are altered. And the benefits of gene therapy is to replace or add new gene in an attempt to cure disease or improve body's ability to fight diseases. And also, it has a promising potential not only in the field of medicine, but in some related fields like agriculture. The next one will be the disadvantages of gene therapy. Modifying organisms means modify its capabilities. It can damage the gene pool resulting to multi-gene disorder, high cost, and ethical issues. So the gene therapy is expensive. It can be extremely pricey, making it inaccessible for some people. And also, the gene therapy has unethical issues to some people. For example, they might be wonder if the use of gene therapy can make them less accepting of people who are different. And also, the gene therapy may cause infection. Some gene therapists use inactive viruses to carry healthy genetic material into a faulty cell in the body. It's possible that these viruses could infect the persons receiving therapy.
Good day. My topic for today is biotechnology. First, what is biotechnology? By the way, biotechnology is a technology that utilizes biological system, living organisms, or parts of development, or create different products. So, what is the use of biotechnology? The use of biotechnology is a biological system, living organism, or derivatives, therefore, to make or modify product or process for specific use. Like instant example of this is as high standard crops, new variety of crops. Then there are two specific principles of biotechnology. So the two principles of biotechnology is the chemical engineering and genetic engineering. First, let's tackle about chemical engineering. Chemical engineering involves the productions and manufacturing of products through chemical processes like fertilizer, petroleum, pesticides, other chemicals that involve chemical processes. So in the other hand, the genetic engineering is the process of using recombined DNA or rDNA technology to alter genetic makeup of the organism. So the use of the genetic engineering is a technique that allows genes and DNA to be transferred from one source to another, leading to the production of living modified organisms or genetically modified organisms. So they will add some genes on that product instances or that crop to have a great production and resistance of pests or viruses. So the purpose of genetic engineering is to help the farmer to have a great impact of their harvest and a big profit of their products. The next topic is the genetic modified organisms or GMO. What is GMO? GMO is organisms whose genetic material has been altered using genetic engineering techniques like plants, animals, organisms, or other organisms whose genetic makeup has been modified using recombinant DNA methods. Example of this is BT corn, 2GM papaya, ground up ready soybeans, golden rice and Arctic apple, and aqua advantage salmon. So, through help of the genetic engineering, we can have new standard crops and maka-add pud sila og nutrient value like the golden rice pataasan niya og vitamin A, and like sa BT corn pod di na tam kailangan mo apply og pesticides kay nana sila own pesticides sulod sa ilaha and dili sila maka-harm sa atuang lawas. Now, let's tackle about the 6 benefits of GMO. First is higher efficiency in farming. Second is increase in harvest. Third is control in fertility. And fourth is increase the food processing. Fifth is improvement of desirable characteristic. Sixth is nutritional and pharmaceutical enhancement. So with the help of GMO, dakan ligay matabang ang GMO sa atong farmer. Mataas ang efficiency sa atong farm, dako ilang ma-harvest og less atoang hatag na fertilizer sa atong crop kay sustainable na pud, og the best is the ma-increase tong food processing. Then ma-improve pud ato ang characteristics sa atong crop, og lastly makahatag ta og higher nutritional value sa atong mga crops. Kung manaman ta sa atong benefits, let's go to the potential risk of GMO. So first potential risk is inadequate studies on the effect of the GMO to the human and environment. Second is promote mutation in organism which the long-term effect are still unknown. Third is human consumption might have the following effect. First is more genetic reaction. Second is gene mutation. Then third is antibiotic resistance. Then fourth is change in nutrition value. This topic is the biosafety on GMO. First, what is biosafety? Biosafety is a prevention of the large-scale loss of biological integrity, focusing both on ecology and human health. So, the use of biosafety is to protect us from the harmful incidents. So, under mga rules ang biosafety, the 3 rules sa biosafety is the Codex Alimentarius Commission. What is Codex? Codex is responsible for the development of standards, codes of practice, guidelines, and recommendation of food biosafety. So, mo ni siya ginasunod sa tanan whole world na ginasunod nila mga guidelines or mga standards na pagdevelop nila sa ilahang products. Next is the Cartagena Protocol on Biosafety. What is Cartagena Protocol? Cartagena Protocol is an international environment treaty that regulates and transboundary of the movements of LMOs. So, the Cartagena, it is a movement of the product. Kung ahagi ka ng product, kung aha pod gi-export ang product, dira pod makita nila kung aha ni gawas o aha ni sulod, para na mga instances na if na mga daot or expired, dali ra nila ma-locate or mapakitan or dali ra nila makitaan kung ahagi ka na product. Example sa China, nag-deliver siya sa Philippines, so nakitaan sa Philippines na naay daot sa China na mga products, so ilalang matrace na dali kung kinsa to na cargo na ilang gidala ay gipadala dito nila. Next is the International Trade Agreement of Labeling of the GM food and food production. Any agreement that required exports of GM food and food production to label their products and give right to importing parties to reject and accept the GM product. So, mo na siyang pinaka-importante. You must check the product if you want, if magpalit ka. First, magpalit mo sa mga grocery store, gina-check na to product para kung kuan ba, kung expired na ba siya or dili pa, or accept pa na to reject mo ra gyapon sa biosafety. Ginatan-aw nila ang mga products na moabot sa isa ka country or ilang gina under sa mga quarantines nang gina-check nila na if dili pa daot, if dili pa expired, o ang kanusa pud na date na gihatod nila ang product para matan-aw sa consumer na kuan okay pa ato ilang gipalit na product. Kay ang consumer kay naman sila right to choose na unsa ilang paliton, og dapat mo tan-aw pud ta sa label sa mga label sa mga product para dili pud ta madaot sa atong kagalingon. So that's all, thank you.